Welcome to The Mixer. I'm your host, Andy Marshall. I'm here with Captain Cocktail himself, Juan Carlos Lopez. How are you doing, JC?
Very good, Andy. Excited to welcome the spring. And before you know it, we'll be ready with the summer cocktails. But before that, I'm very excited about the conversation today. So who is our guest?
Yeah, the man in the hot seat is Robert Pacifici. He's the Chief Scientific Officer at CHDI Foundation, which is one of the leading non-for-profits with a mission to bring a Huntington disease therapy to market. Robert is someone who I think one would call a veteran of the drug discovery business. Before he came to CHDI in 2003, he was CSO and site head at Eli Lilly's center in Research Triangle Park in North Carolina. Before that, he was at Xencor and Amgen. So we're really looking forward to talking to him about some of the latest developments in Huntington's.
Yeah, I'm really looking forward to hearing particularly about what went down during the recent Therapeutics Development Conference in Palm Springs. They hold that conference every year, and it seems that this year was a particularly exciting occasion.
Yeah, they had over 400 researchers attending this year, I think, and there was a real buzz not only in terms of several companies initiating clinical programs that seek to take forward some of the approaches to downregulate Huntington protein, but also that there was a raft of startup companies and posters concerning drugs that are targeting an entirely new mechanism, mismatch repair. So we're going to be hearing a bit about that in the following podcast. So looking forward to it.
Excellent. Let's get started.
Let's get stuck in. Robert, a very warm welcome to The Mixer. Thanks for coming on the show.
Thank you so much for inviting me. I'm delighted to be here.
Yeah, so we were very excited to bring you on to talk about some of the latest developments in drug discovery around the neurodegenerative disease of Huntington's, of course. But perhaps to start things off, you could tell us a little bit about your background, how you ended up at CHDI, the Foundation's mission, and talk a little bit about your work in taking that mission forward.
Happy to do that. Kind of depends how far back you want to start.
Well, it's been a while. You've been there, what is it, over 2 decades?
I've just celebrated my 20th anniversary. And if you wanted to go back even further than that, I suffer from a rare genetic disorder myself called familial Mediterranean fever. I was very sick as a kid, and I moved to Italy and got a new pediatrician who looked up the disease. He was unfamiliar with it and called us immediately that evening and said, "Were you aware that there's a new finding, that there's a treatment for this disease?" So I started taking a pill the next day, and it's now been 52 years, and I've been completely asymptomatic. So I got to experience firsthand the wonders of drug discovery. Went from being on a first name basis with all the school nurses to now being able to participate in sports and leading a regular life. It was transformative for me. So I always knew that it would be great in some way, shape, or form to pay that forward. And I always had an interest in problem solving, puzzles, science. And so my career was always laser focused on getting to where I could do some of these things, and had the opportunity to get trained as a biochemist, both undergrad, UMass, and then a PhD in biochemistry from USC, and went on immediately to do my postdoctoral fellowship in an industrial setting. So I went to Amgen for what was supposed to be a 6-month internship and ended up spending a decade there, learning all about the biotech industry and about drug discovery. Transitioned from there to a small biotech called Xencor, which did rational protein optimization, and then had the privilege of working at Eli Lilly and Company to get my big pharma stripes as their site head, and down in North Carolina at Lilly's RTP site, and as head of global quantitative biology. While I was there, a gentleman by the name of Roby Blumenstein, who was the president of CHDI. We met actually at an NIH symposium about drug discovery. And he explained to me a little bit about CHDI Foundation, which I like to characterize as kind of a magic, tragic coincidence. Mm-hmm. So what I mean by that is he explained to me what a horrible unmet medical need Huntington's disease is. It really is in every way, shape, or form. That's the bad part. The magic part is, as you know, there are large numbers now of very generous private donors who are interested in funding biomedical research, including Huntington's disease. And so the convergence of the resources and the singularity of focus and purpose became very attractive to me. Yeah, I made that transition about 20 years ago, and it's been just a remarkable journey.
Maybe we could also talk a little bit about the CHDI Foundation's mission, because it's particularly focused on this idea of collaborating with all of the different stakeholders in drug discovery and development. Could you kind of expand a little on that?
Yeah, of course. So it really is, as you say, a unique organization. There are quite a few other voluntaries now, Michael J. Fox, the Spinal Muscular Atrophy Foundation, that are similar. So there are certain characteristics about CHDI that are, I think, important to highlight. The first one is indeed that we are completely foundation funded. So we have the support of very generous private donors, and it's very significant support. So CHDI spend is on the order of about $100 million per year. So this is a very significant effort. And those funds come to us without some of the traditional burdens of fundraising, of going to granting agencies, of going to many smaller donors. So we can really focus on the science. As a consequence of having donors instead of investors, we enjoy a not-for-profit status. So what that means is that we only have collaborators, no competitors. So if something I say today causes somebody that's listening to this podcast to find a treatment for Huntington's, terrific, we win. Mm-hmm. That's what we're all about. And it really allows us the luxury of the singularity of focus. So we have a fiduciary responsibility to our donors to stay focused on Huntington's disease. There are many organizations, ones that I've worked for, that say they're interested in one area or another, but because drug discovery is so hard, when they gain traction in another area, they have to follow where the success is. We have this fiduciary responsibility to make sure that we stay focused on Huntington's, and the mission of the foundation is to accelerate therapies, meaningful therapies for Huntington's disease. So we chose each one of those words carefully, accelerate, to indicate that we're a time-motivated organization. Our bottom line is time, not money. It is exclusively Huntington's disease. So if we discover something that might be good for something else, we're happy to give it away, but that's not part of our remit. And meaningful therapies, because, pardon the baseball metaphor, we're not looking for a bunt or a single. We're looking for a home run. In other words, we're not looking for palliative or symptomatic treatments. We're looking for disease-modifying treatments, something that significantly moves the trajectory of the disease in a positive direction. We typically avoid the word cure because there are precious few diseases that are cured, mostly infectious diseases, but you get the idea. So the foundation funded is one aspect of it. The second aspect, as I said, is the singular focus on HD, which allows us a time horizon that's maybe different for other drug discovery organizations. When I worked at Lilly, we had therapeutic areas in other unmet medical needs that when they proved difficult or when there was a change in personnel were abandoned. And the reality of it is that drug discovery takes 10, 15, 20 years plus. And so you need that kind of continuity and those kinds of resources over a protracted period of time. Our role as collaborative enablers is really key as well. So how do we do that? Well, in a variety of ways. Let's not mince words. One of the ways you enable people is you pay them. And so CHDI has served as a fairly significant funding agency, mostly for academic researchers. So we don't do grants, we do contracts, which allows us to make sure that the money that we're providing is indeed spent on the specific scientific aims that were articulated at the beginning of the program. It also ensures us that once there's success in a particular area, that we're able to reap all of those benefits and share them with the rest of the community. So if you develop a mouse model, we can share it with everybody. If you develop a compound, we can share it with everybody. If you develop a protocol or a cell line, those are things that are shared with the rest of the community so that it floats all boats, so to speak. And we play a big role in facilitating not only tools, reagents, and models, but the data that goes along with it. As you're probably very familiar, having given your backgrounds as editors, there's a huge bias, success bias, in terms of what actually gets published. Well, all of the data that's generated that turns out to be negative is still valuable. In some cases, to dissuade people from repeating something that's already been tried and failed. And in other cases, because those data might be useful for somebody else in a different way. Maybe they want to reprocess it. Maybe they want to concatenate their data and add to it to achieve statistical significance. And so a big part of our platform is also to make sure, in addition to the more traditional scientific publications, that we're getting information, data, and knowledge out to anybody that's interested in the types of problems that we're solving. And we do play an integrative role as well. So matchmaker, right? How do we bring people together so that your peanut butter and his chocolate come together for a great Reese's peanut butter cup? We still very much view ourselves as a scientific organization. So CHDI has adopted what we sometimes refer to as the virtual model. I don't like that word so much because it sounds like we're fake and we're very real. So there are now almost 140 people who work within the four walls of the foundation across our 3 sites, New York, Princeton, New Jersey, and LA, where I'm based. But if you came to visit us, you would not find any clinics, you would not find any wet labs. We do everything on an outsourced basis. So on any given day, in addition to the people who work for CHDI itself, there are about 750 people who work around the globe on our behalf. And we think that this model is particularly suited for our problem because it allows us in a very fungible way to engage the best and the brightest in any sector, in any part of the world when we need them to help solve a problem. And then without rifts or layoffs, we're able to reconfigure the resources that we have at our disposal to solve whatever the next problem is that we're facing. So we have contract research organizations, we have our collaborative network with academics, and the thing that I'm particularly proud of, we'll talk more about this later, I'm sure, is engaging the biopharmaceutical companies that also bring critical resources to drug discovery. When I joined the foundation, there were exactly 0 companies working on Huntington's disease. Since then, there are now over 60 companies that attended our conference recently in Palm Springs. Now, some of them, it may be nothing more than a one-liner on their website that says Huntington's disease. Who knows what they're really doing? At the other extreme, there are companies like Genentech Roche that have spent triple-digit millions now with their clinical candidates, and everywhere in between. So it's been very comforting and rewarding for us that CHDI has been able to create a landscape that is fertile enough for companies now to want to train their very big guns on Huntington's as an unmet medical need. And we believe strongly that in addition to Huntington being a big win in terms of an unmet medical need, there's also, I think now, a very clear pharmacoeconomic validation that these rare and neglected diseases are worth having in your portfolio, even if you're a for-profit company. And Huntington's may be the best example of a neurodegenerative disease that is worth working on first, given the fact that it's a monogenic disease with 100% penetrance. And so we have some advantages in connectivity from genes to cells, from cells to animal models, from animal models to people, that may make it a little bit easier for us to deconvolve some of the intricacies of neurodegenerative diseases in a way that then serves as a heuristic for some of the larger market cap diseases like Parkinson's and Alzheimer's. So I think that's been part of where the interest has come from as well.
Maybe we could talk a little bit now about our knowledge, understanding of the disease itself. Although Huntington's is often held up as this example of a archetypal dominant gain of function genetic disease, as you said, with 100% penetrance. If you find you have this expansion mutation, you will get the disease. At the same time, it's a really interesting example of, in some ways, the scientific hubris often associated with simplistic explanations of human biology. Yeah? Because what do we know today about the protein encoded by the Huntington gene? And what do we understand about its role in pathomechanism?
Your comment resonates with me 100%. And I think, we have to substitute hubris with another H word, which is humble. I've been humbled by the complexity of what seemed to be a fairly simple disease. And I think that pursuing tenaciously at a high degree of granularity, an understanding of what's broken before you try and fix it is critical. I would say the way to do that is by studying the one and only beast on the planet that actually gets Huntington's, which is humans. So we start our journey there. And as you correctly point out, the sort of first sentence is Huntington's is a fully penetrant, dominant disease that is caused by a CAG expansion in exon 1 of the Huntington gene. Okay, so that's step one. But this degree of granularity is really important. So most recently, what we've learned is that it's not even just getting to 40 CAGs, which is the size of the expansion, which has served as the threshold for actually having the bad gene. We now know that it's an uninterrupted CAG. So if you have another codon in there, even if it codes for glutamine, would make the same exact protein, this inverse correlation between the age of onset and the size of the CAG now needs to be amended to say the size of the uninterrupted CAGs. So it's really important, just underscores this notion of being very precise in what we think we know and what we don't.
This is an absolutely key point, yeah, is that with our kind of traditional idea that this is a polyglutamine-driven disease that leads to these aggregates. And yet, if you have these repeats and they're broken by these interruptions, you get these differences.
Yeah.
This is a kind of profound insight, yeah?
Yeah, and I'm not trying to avoid your question. I am embarrassed to say, on behalf of the community, that here we are some 40 years after the identification of the marker and 30 years after the cloning of the gene, we still don't have a satisfying answer as to what the Huntington protein does. There's certainly a lot of very interesting insights about scaffolding, about binding to DNA, about other very intriguing things that the Huntington protein might do. But from a therapeutic standpoint, one of the things that we've really been focused on now is we don't even really know for sure where in the central dogma things actually go wrong. In other words, it's formally possible that it's just the expanded DNA that's the problem. There are certainly forms of the expanded RNA that adopt different conformations. Maybe that's problematic. It's certainly precedented in other diseases like Duchenne's muscular dystrophy. Certainly, there's been a lot of focus on the protein itself, given the fact that other neurodegenerative diseases share these misfolded neurotoxic proteins. And those three possibilities are not mutually exclusive. It could be all of the above. If you go further into the protein itself, there's been a lot of discussion on exactly what proteoform. So is it a misfolded monomer? Is it an oligomer? Is it fibrils? Is it aggregates? Is it aggregates in the cytoplasm? Is it aggregates in the nucleus? There's actually, and we'll talk about this maybe as we get to some of the very cool presentations that were made at the 20th HD Therapeutics Conference that we just hosted in Palm Springs. There's a lot of focus recently on a misspliced variant of the Huntington gene. So it turns out that the larger the CAGs, the higher the propensity for a missplicing event, which generates a short exon 1A fragment that just has the first 17 amino acids and then the poly Q stretch and nothing more. And maybe that is the toxic species. Maybe it's the seed that causes the rest of the protein to aggregate. And so there are now interventions that are in the clinic that differ as to whether or not they would lower Huntington in an allele-specific way. So is it both the wild type and the mutant? Some of them will lower Huntington 1A, some of them won't. And so trying to get out ahead of understanding that so that the target product profile for future therapeutic interventions can be tailored to maximize success is really important.
To me, this is very interesting because I remember when I was in grad school, the gene had been cloned and everybody was very excited about that. And then there were these classic papers that came out in Cell. This was when conditional knockouts were just being started, right? And there was this paper by Yamamoto and others showing that if you reduced the production of the mutant Huntingtin, even if there were aggregates, the aggregates dissolved, and then you had a mouse that was perfectly healthy again. And I think that sent the field searching for that solution for a long time. The people invested a lot of money and a lot of time because they felt that simple explanation was going to be the winner. And now we know that that wasn't the winner, that there are still a lot of things to know about the biology. And thinking from the drug discovery perspective, I wonder what your thoughts are about where exactly the problem for developing therapeutics for Huntington lies, right? Clearly, the biology is something that we haven't really understood. But thinking in terms of the tools and the prerequisites that you have to really get into proper drug discovery, do we have the right tools for this problem? Do we have the right animal models? Do we have the right natural history studies? What may we be missing? Because clearly, it's certainly not a simple disease. We can say that very categorically. But considering all the understanding that we have, all of the data from animal models, this cohort in Maracaibo, Venezuela, right? That was another big boon for this field. To me, there are like a lot of things in place, and yet it continues to be problematic. So I'm trying to see if you think that there's something beyond the biology itself that we're still missing that may catalyze drug discovery in this field.
Yeah, it's a very insightful question and certainly falls squarely in the remit of CHDI to identify what these potential bottlenecks are and to remedy them. Let me answer the question in a couple of different ways. The first thing does relate back to the biology itself. As you correctly point out, Yamamoto's paper was very transformative for the field. Certainly the idea that a monogenic disease is caused by a single gain of tox gene makes sense as a therapeutic strategy to turn it off. There are now no fewer than 6 clinical stage companies doing that. Terrific. I hope they're successful, and that would be great. But what we knew fairly early on is that this inverse correlation between age of onset and CAG length only accounts for about 65% of the effect. We further know that the remaining 35% is not environmental. So in other words, the tendency to be an early onsetter, earlier than the mean or later than the mean, is also heritable. And so one of the tools, to your point, that was missing was the ability to find these modifier genes. Well, Huntington's is a rare disorder. Modifier genes are even more rare on top of that background. And so one of the things that CHDI endeavored to do right from the get-go is establish what I think, and I wasn't involved in this, so I can be very positive about it because my colleagues put it together. But Enroll HD is a prospective registry trial where now more than 20,000 persons with HD and family members are enrolled so that we can take biosamples from them. We're actually doing full genome sequencing of them. We actually have longitudinal data where they come back year on year. And this allowed, among many things, the launching of what ended up being 6 consecutive cumulative genome-wide association studies that allowed us to identify many modifier genes that contribute to this heterogeneity. In a remarkable convergence of biology, what we found in the genes that finally met genome-wide significance in those GWAS studies, half a dozen of them all shared a biology in that they were involved in DNA mismatch and repair. We further were fortunate to find variants that pushed in both directions. In other words, variants of the same gene that made the onset later, variants of the same gene that made the onset earlier. So both genetic pharmacologies, if you will. The last bit of good fortune came from the fact that we were able to now make the mechanistic hop from DNA mismatch repair to the mechanism of somatic instability. So this was the second sentence. You need to have the Huntington expanded gene. And now we know that you have to have somatic instability. So it is formally possible that having 40 CAGs, while necessary, is not sufficient. In other words, it may be that even a protein with 40 glutamines is not toxic. And it's only over a period of time that it expands. This is the work of Nat Heinz, Steve McCarroll, and now Don Cleveland will join in characterizing postmortem human brains to see that actually what's going on at an individual cell level in the organ of interest, the human organ of interest, over time. And so I've never seen in my career a more definitive and accelerated version of understanding new targets with human validation, having a mechanistic logic link, and launching into interventional therapeutic programs as quickly as the culmination of those 6 GWAS studies that were done with 9,000 participants across a consortium that leveraged the Enroll HD platform. So that platform is a critical tool, not only for these types of mechanistic studies, but also for the interventional studies, because pharma companies now can come and query that database to find all of the participants for their trials that meet the inclusionary and exclusionary criteria and have all of the run-in data from the longitudinal aspect of it that allows for these much more robust clinical studies. The other area that I would point to in terms of tools and what's missing is biomarkers. Nobody is going to the clinic anymore to enroll hundreds of patients over many years, keeping their fingers crossed that they're going to move a clinically meaningful super score. People want a signal earlier. They have to have a signal of safety. They have to have a signal of pharmacokinetics, that they're getting exposure. They have to have a signal of target engagement. They have to have a signal of pharmacodynamic effect, that the compound is doing what it was crafted to do in the right place at the right time. And ideally, some signal of biological efficacy. Now, that doesn't mean that it's guaranteed that you will matriculate all the way to a clinically meaningful benefit. But what it means is you're taking a real shot on goal. And what it also means is that if that shot on goal fails, you will learn from the experience in a way that will inform subsequent drug discovery efforts. So I think that those are incredibly important elements that have been missing and that CHDI is trying to provide to the field.
Robert, I kind of want to segue from what you just said there into also some of the recent work that came out of the McCarroll lab. I'm interested as to how CHDI is thinking about some of that biological insights that came out of the McCarroll study, which essentially was using single cell RNA sequencing, combining that with long read sequencing to look at how these CAG triplets expand over the lifetime of an individual. And then there's this critical threshold, that they found is reached at around 150 repeats. Maybe you could tell our audience a little bit about what was found in that study and how you're thinking about it.
Well, it's been an incredible set of findings, and it's enjoyed quite a bit of notoriety, including at our conference. We've had now for 3 successive years, people, including Steve himself, presented at our conference, because one of the things I'm fond of saying is there's nothing more precious to a drug hunter than an observation made in the population you seek to treat. Well, that turns out to be a tall order. Experimental medicine trials are often ethically constrained, expensive, inconclusive. Interventional trials, even more so, and even fewer and far between. And so without giving up on this notion that we have to study people, there's been a couple of really celebrated examples. The genetics that I just talked about in the GWAS, and now this utilization of novel technologies to look at postmortem human brains, including Steve's work. So the incredibly exciting thing that Steve found, as you indicated, is that there are these different phases, it appears. Asynchronous, by the way, so it's not like the whole brain is in lockstep, and especially in medium spiny neurons, which we know are the cells that seem to be the most vulnerable to the Huntington insult, seem to be the ones that not only have potentially an accelerated rate of somatic instability, but a resilience to smaller expansions and an aversion once they reach this particular threshold, maybe 150 or more. If that's true, it has some pretty important consequences. It explains maybe some of the time domain that we see. Why is Huntington's a late onset disease? Perhaps it allows a larger window during which treatments can be administered, right? So one of the worries was that if people are treated late in the disease, that it would be too late, there would be no opportunity for therapeutic rescue. So it gives some hope on that front as well. But it also, back to our discussion about granularity, it starts to allow us to understand what the real insult is and allows us to start thinking about the third leg of the stool, which is what are the toxicity drivers? So once you have an expansion of that size, what is it that that particular expanded DNA, RNA, or protein is doing to screw up cellular function in a way that translates into the pathophysiology, and in a way that if we could reverse it would result in a clinically meaningful outcome, making people feel, function, or survive longer. So it's been an incredibly impactful set of studies. Now, it's not without its detractors, not surprisingly. And so there's an awful lot of more work that needs to be done to help characterize these initial findings. And there are other labs, as I said. Nat Heinz's work in this area has been pivotal. And recently, actually, the featured speaker at our HD Therapeutics Conference was Don Cleveland, who is rejoining the fight. He was involved in HD research very early on and collaborated with CHDI very early on when the first Huntington lowering therapies from what was then ISIS, now Ionis, developed an antisense oligonucleotide to lower Huntington levels. But he's now developed a novel platform that allows for a different type of characterization, again at the single cell level. And we hope that that will complement with additional data. We're also bringing single cell and spatial proteomics into the mix. And so all of those things taken together will hopefully give us the additional granularity that we need to figure out whether or not Steve's observations hold up, exactly what the downstream consequences are, and how that would influence our desire for different types of therapeutic interventions and what the target product profile for those would be.
One point that I found really intriguing about the finding is, Steve says it's stochastic process, this repeat expansion. So even in late stage disease, you only see a if you're looking at any particular brain region, even the striatum, there's only 10 or 20% of the cells that are actually at this kind of repeat threshold.
Right.
So if you think about the implications of that, that means that the rest of the cells that haven't yet crossed the threshold potentially could be helped by some kind of therapeutic intervention via a kind of mismatch repair inhibitor. Or do you think that's reasonable?
That's very reasonable. I think that since the cloning of the gene itself, there's nothing that has been more impactful for the field than the discovery of these mismatch repair genes as human validated points of intervention. So the field has responded really well to this. I think that the combination of these different phases and the window of opportunity to slow down somatic instability in vulnerable cells, and the fact that we have a handle on the enzymes that are responsible for that yin and yang balance of accelerating rates of somatic instability, MSH3, so let's inhibit it, of slowing down rates of somatic instability, so let's foster that, FAN1, are great examples of handles that we now have as drug hunters.
We have known the gene for 30 years, as you pointed out, and I'm wondering what the state of play is in terms of pre-implantation diagnosis. Because you would have thought that knowing the gene so well would lead to a decreased prevalence, and that over time people would have already been on board with Huntington's. And yet we still have a large population of patients. So I wonder if you know of any explanation for that. How is it that pre-implantation diagnosis hasn't really made up a difference as substantial as one might expect?
For people unfamiliar, because we now know the gene, it should be very easy to say we're going to do pre-implantation genetic diagnosis and only implant embryos that don't harbor the mutated gene. And this works and has been used wonderfully by many families. Why is it not maybe as widespread as one would have predicted? The answer to that is a little bit complicated. The first is economic. You mentioned the incredibly precious contribution that the Kindred and Lake Maracaibo in Venezuela made to the cloning of the gene.
Yeah.
I think it will be a long time before a pre-implantation genetic diagnosis makes it into countries like that. Even here in the States, where maybe people have the economic resources to do it, it's further complicated by a couple of things. The first one, as you probably are aware, is that there are what appear to be sporadic cases of Huntington's disease. So there's a phenomenon called anticipation. There are families that might harbor a gene that is subthreshold, maybe 36 or so. And then because the gene can expand through the gametes, especially when it's paternally inherited, all of a sudden now you have a new Huntington's family where the kids inherit genes in the 40s and are going to get the disease. Those families would have had no way a priori of knowing that they should have been tested and undergone pre-implantation genetic diagnosis. The third complication is related to the disease itself. I'm not a neurologist, I'm not a psychologist, but we know that there are many aspects of Huntington's disease, including a denial that people have them, including a high rate of adoptions where it's not clear that the adopted baby came from a Huntington's family. And in all of those cases, it would obfuscate the ability for someone to know that they should engage in pre-implantation genetic diagnosis.
Another thing that to me has always been interesting in this space has to do with regulatory science, because it's beautiful biology from the point of view of knowing that if you have 40 or more repeats, you will develop the disease. And granted, that accounts only for 65%, and there are the gene modifiers, but 65% is pretty good. So because of that, one would have thought that the regulatory guidelines for developing therapeutics for this condition would be in a way more generous, and that people would be allowed to do prospective trials and try to help people before they develop the disease. And I'm not sufficiently familiar with this field, so I wonder if you can educate us and our listeners in terms of what is the state of play with the regulatory science in this space. And before you start, the other thing that to me is relevant in this context is this issue of the disease modifier, because those molecules will have to be tested also in these patients. And how long would a trial have to be for something that is not going to be targeting the direct mechanism, but more modifying the pathogenic mechanism? It may require a pretty long trial. So if we're serious about that therapeutic route, I wonder what the regulatory landscape is going to be for that kind of therapeutics.
I'll answer the question with two big caveats before I start. The first one is that my wife is the chair of clinical, regulatory, and quality sciences at USC, and we respect each other in that. That's her end of the pipe, and I'm a preclinical guy. Now, the second caveat is that CHDI is very fortunate to have a chief medical officer, Cristina Sampaio, who handles the clinical end of things. She is an MD, PhD neurologist, and she actually was a regulator for the European Medicines Agency and is incredibly facile in these matters. What I can tell you as a preclinical scientist is that you're absolutely correct that if the gambit is strictly running a traditional trial that requires a clinically meaningful outcome, this is incredibly difficult, and we should be able to leverage some of the things that you said. The fact that we know who the gene carriers are, the fact that we could come up with our version of a polygenic risk score based on the modifiers that they have. And CHDI has been playing a role within the HD R&D landscape to interface with regulators to push the idea that a combination of not yet surrogates, but biomarkers taken together should be good enough, at least for conditional approval, even if follow-up is required until you hit your clinically meaningful outcome. And so I would not be surprised, and I'm very hopeful, that in the near future, a combination of things that ensure you've got a pharmacodynamic response. For example, I can measure that Huntington is being lowered, and CHDI was critical in developing the bioassay that's used in cerebrospinal fluid to measure the diminution of Huntington after an intervention. We're working tirelessly on the development of a mutant Huntington PET ligand so that we can add to that with spatial and temporal resolution, which are obviously not getting in CSF. But the combination of a pharmacodynamic biomarker along with some measure of efficacy, things that have been adopted more broadly in other neurodegenerative diseases like neurofilament light chain, for example, which has enjoyed a lot of success in other neuro-D indications, that hopefully that combination is good enough to get at least conditional approval so that we can overcome some of the longer timeframes that are required to hit clinically meaningful outcomes. I think that that's where the field is going. I think it's appropriate. I think that after the first few things actually do meet their endpoints, so the way trials are being designed now is using another big tool that CHDI developed, which is the HDISS, the Huntington's disease staging system. That allows for much more robust, much more ubiquitous and well-defined staging of people across the disease. And so as people adopt this now, we can do the current trials at the precipice of people becoming symptomatic, early symptomatic. And that's now much better defined in the HDISS. And that once a few of those interventions are able to move the clinically meaningful needle, then all of the things that went into, all the measurements that went into making those trials hopefully will then serve as more robust biomarkers and maybe even in the future surrogates of efficacy for future trials.
Super interesting, Robert. I mean, there are also I think Wave made some announcements at the end of last year that the FDA is increasingly kind of receptive to this, yeah?
Yes. We didn't get a chance to talk much about the recent HD Therapeutics Conference, but one of the sessions that we had at the very beginning, sort of session 0 before we got to the regular translational program, was an update from each one of the companies that are at clinical stage, which for me was very gratifying. When we started this conference, nothing was in clinical trials, nothing meaningful other than thermonuclear doses of nutraceuticals. So it was great to hear that these very credible companies with very credible, well-designed clinical trials were reporting out on various advancements. And yes, we asked companies like Wave,
Unicura, yeah.
Yeah, to give us a bit of a heads up as to what the nature of their interactions with the FDA was in granting them some leeway in the hopes that that would apply to some of the other trials that are ongoing.
It would be great to just talk a little bit about how we see this clinical development field opening out over the next few years, Robert, because in some ways one can see with Huntington's that there are some resonances with what we see in other fields. So, as you said, in some ways the clinical development programs have moved into earlier patients. And as you said, the HDISS staging tool has been very important in that. Some of the things that I've heard Sarah Tabrizi talk about at UCL are ultimately where we want to get, maybe even into the prevention arena, where you're talking about patients, individuals who have no symptoms, so pre-manifest patients, and one would be treating them and pushing out this threshold at which one starts to see the toxicity and symptoms actually kick in beyond an individual's lifespan. Could you talk a little bit about how you see all this developing, going from where we've been, which is treatments of last resort, to actually treating people earlier in disease, and the prospect, which I think the whole entire Huntington's community would love, is this idea that one may get to a stage where one could be taking a particular intervention and push out symptomatic disease to the end of one's life.
Right, so I started this discussion at the beginning of the podcast by telling you about my own journey with FMF. So the gene has been cloned and it's prevalent in Arabs and Sephardic Jews. I married a Korean woman, not because I wanted to dilute my gene pool, but because I loved her. And yet we had a son who started displaying symptoms. I was not hesitant at all to have him tested because I knew that there was a therapy. So in Huntington's, despite the fact that we have a genetic test, fewer than 10% of folks at risk actually make use of that because there's nothing to take, right? So, of course, where we want to land is a situation where people are more than happy to get tested and then more than happy to take a safe and well-tolerated treatment as early as possible. In terms of how we get there, right, that's a little bit more challenging. My one and only visit to the Food and Drug Administration, I used the word prevention. It was on my business card, Prevent Huntington's Disease. And the regulator, I'll skip his name, scolded me and said, "Son, you can't prevent Huntington's disease, you're born with it," because he meant that you have the gene. And so once you've got it, it's more a matter of treating the disease. It's a matter of how early you're treating it. So you're right. We want to be able to get to a point where we have the types of interventions that we know will work at an early stage. We know that are safe and well tolerated. I mean, as you can appreciate, folks that are, we're not supposed to use the word pre-symptomatic anymore either, by the way, because of HDISS, but people who don't manifest any of the traditional signs and symptoms look pretty darn healthy. And so you can imagine that the bar is set pretty high. Even though it's a fatal disease, you certainly wouldn't have large uptake for an incredibly invasive procedure or an incredibly toxic procedure, early stages in the disease, as you would for a chemotherapy for oncology. So we have to think very carefully about how that plays out. So my strong sense is that what the future holds is we still have not seen that we can move the needle in people in a clinically meaningful way. That will happen. And when it does, then we leverage off of that to say, what other co-therapies are going to be necessary? How do we improve the intervention so that it's more potent, it's more bioavailable, it's more selective, it's easier to take? And then how do we move that as early as possible in the disease trajectory, ideally so that we never have what we would refer to in the past as traditional disease onset.
I have a couple of final questions. One of them has to do with one of my personal interests, which is these insights that one can have from ultra rare diseases into how they can help us understand common diseases or other forms of rare diseases. And in the case of polyglutamine disorders, as you know better than I do, there are a lot of polyglutamine diseases that are extremely rare. Maybe a dozen families have been identified for some of them. And mechanistically, yes, there will always be differences because each gene is different. But the fact that they share these polyglutamine expansion at the core has led some people to believe that if you find a treatment for one, maybe that will bode well for other conditions, right? And they can be either the same therapeutic, it could be a platform technology, et cetera, et cetera. So I'm wondering what your impression is of that possibility. Do you see insights from ultra rare diseases helping develop therapeutics for Huntington's, or vice versa? Do you see advances in Huntington's that could be taken to the clinic faster by testing them in an ultra rare population of patients?
Absolutely, yes. We refer to this phenomenon as adjacencies.
Yeah.
So what we're looking for are what are the adjacent diseases on any dimension to Huntington's? What are other diseases that are monogenic? What are other diseases of the striatum or medium spiny neurons? What are other diseases that key on a neurotoxic unfolded protein? What are other diseases that share the commonality of a triplet expansion? And so, of course, we want to help each other bidirectionally. If there are things that we do that could help these other diseases, terrific. If there are examples, as you say, because they might be afforded certain liberties in ultra rare diseases that we don't, but they can generate the human proof of concept, terrific. Absolutely. And I believe that that mechanistic logic link is rock solid. So I think if we develop an inhibitor for mutas beta, which is one of the enzymes that's involved in driving somatic instability for Huntington's disease, I have every confidence that the very same compound could be applied in other diseases that share the same mechanism of somatic expansion.
Yeah, that would be really incredible. And I think it could work the other way as well, right? If you have these compound, or if a company develops this compound, they can have faster access to market by testing in an ultra rare population, get an approval, get an increased valuation, and then have the cash to do the Huntington trial.
You're absolutely right. In fact, we shouldn't limit ourselves to where that advantage can come from. If their particular gene is peripherally accessible and therefore easy to measure than the brain expansion, terrific. If their gene expands more quickly and therefore the time of treatment needs to be shorter, terrific. So all of those things are things that we should be exploring tenaciously.
Robert, this has been very insightful. We've enjoyed a lot our conversation. But before we let you go, as you know, at the Mixer, we have a passion for cocktails as much as we have a passion for science. So we're hoping that as a farewell question, you would indulge us sharing with us what is your go-to cocktail after a particularly productive meeting like at Palm Springs and you want to chill out and enjoy. What is your go-to drink?
I'm Italian by heritage, but I've spent most of my life here in the United States. So the convergence for me is a drink called the Boulevardier, which is basically a Negroni, but instead of gin, you use American whiskey or bourbon. So this is one of my favorite drinks that actually I tried for the first time in Palm Springs, where we hold our yearly therapeutics conference. It was at the Copley, which is Cary Grant's old place. And this old-time waiter, I asked him for a new suggestion. He suggested the Boulevardier, and I've been hooked ever since. It's a very nice combination, especially before dinner, where the Campari is an Amaro that opens up your appetite. And then the whiskey adds enough of a punch so that you're off for a good meal. I love mixology. I love to make them myself. I'm a big fan of Angel's Envy, which is the bourbon that I use. It's a bit of a play on words, right? So when you make whiskey, some of the whiskey evaporates. They call that the angel's share. The part that stays in the barrel, that's the Angel's Envy. So it's a great combination.
Very good. Yeah, I'm familiar with that whiskey. And so you prefer it with bourbon? Because I have come across people who prefer it with rye as opposed to bourbon.
That's a very good point. You're obviously very knowledgeable here.
Well, it's a mixer. we have to be knowledgeable.
Interesting you should bring it up. So Angel's Envy makes a rye as well. Their rye is particularly strong in flavor because they age it in a rum cask as a secondary aging. And so in a Boulevardier, I might cut it in half rye, half bourbon, so that you hit the sweet spot.
Very good. That's very insightful. When I make a Boulevardier at home, I normally go for rye, I must say. Anyway, Robert, thank you very much for your time and for your insights. It's been a fantastic conversation.
Thanks so much, Robert. Really appreciated it.
I enjoyed it as well. Thanks so much for the opportunity. And if there's anything I can do, don't hesitate to reach out and we'll be very responsive.
Thank you so much.
Thanks a lot.
Well, Andy, I really enjoyed that conversation with Robert. He clearly is passionate about this field, very informed. And to me, it's quite remarkable that 30 years after cloning the gene and thinking that we were on the cusp of solving this problem, we continue discovering new biology, new findings, and it's as problematic as it was 3 decades ago. So it's rather humbling in a way, even though it's nice to acknowledge all the progress that has been made. But I wonder what you thought, Andy.
Well, I believe that biology is always humbling, and as we mentioned during the conversation, there's always a great deal of hubris about how much we understand human biology, and this is a case that illustrates that really well. My take home from this conversation relates to how CHDI has really galvanized this area and has gone about its business in a truly systematic way, and really put together a set of initiatives that I think are really important. One could think about what they've done in terms of corralling patient resources that will speed the recruitment of patients into trials, understanding where patients are, staging disease. And the third part of that, of course, is all of the work that they've been doing to try and forward the markers to measure pharmacodynamics, to look at target engagement, to predict clinical outcomes. All of this is really, really important work that CHDI have been instrumental in.
Yes, it's an example of effective philanthropy. They haven't stopped at just the point of funding research, which is, of course, very important and the beginning of everything, but they have thought about it in a very comprehensive way and making sure that they support everything that you need to launch a successful drug discovery program. So they certainly are an example that other organizations would do well to pay attention to.
So what about the Boulevardier, then? What are your thoughts about the cocktail?
Yeah, I was very pleased to hear that Robert is partial to that drink. As you know, I am a great Negroni aficionado, and the Boulevardier is a close cousin of the Negroni. As we said during the recording, the main difference is that the main spirit is bourbon in the Boulevardier as opposed to gin in a Negroni. But the other two ingredients, the sweet vermouth and the Campari, are the same. You prepare it the same way and you enjoy it the same way. Not exactly a summer cocktail, Andy, but today is not exactly warm. So if you don't mind, I'll go and fix myself a Boulevardier as soon as we wrap up here today.
Sounds good. Keep one for me as well. I think I'll be coming over to enjoy that.
Very good.
So we'll put all of that information about the Boulevardier down in the description of the podcast. Thank you to Robert for an informative conversation. Thanks, JC. As always, we look forward to everybody joining us next time. You can follow us on Twitter. Our handle is at the Mixer Pod, so you can get updates on newly released episodes. And we look forward to seeing everybody next time.
Cheers, Andy.
Cheers, JC.